Thank you. Good morning, everybody. So my name is Ophélie Fauvet. I'm from INRAE, France, and with Daniel Graeber, who will reach us this afternoon online. We are in charge of this session 6, entitled Linking Critical Zone Processes to Biogeochemical Patterns at the Catchment and Landscape Scale. So mysterious title. And it's my pleasure to introduce our keynote, Dr. Julia Knapp from Durham University, where she is associate professor at the Earth Sciences Department. Quick words about Julia's career. She conducted a PhD thesis and some postdoctoral work at the University of Tübingen in Germany, and then conducted some postdoctoral research in Zurich at ETH. And her research topics are related to catchment hydrology, groundwater, surface water interaction, and the quality of water. So for that she used several tracers, such as water stable isotopes. And I will give you the floor, Julia. Be welcome, and the floor is yours.
Well, thank you, Ophélie, for the introduction. Thank you all for being here, and thank you for inviting me to talk here. And yeah, I'll talk about water quality, not about stable water isotopes today, because there's only so much time and I don't want to bore you to death. This all is, well, part of the session, linking critical zone processes to biogeochemical patterns at the catchment and landscape scales. And I think it's actually a really, really cool session title, but I had to think about it for a little bit of what it means, because, I don't know, there's about 150 people in this room, I'd say, and if I asked you what you define as critical zone processes, we probably get 150 different answers. And I think that's the beauty of this, and I'm not a biogeochemist, even though I speak in the biogeochemistry session. I'm a catchment hydrologist, and I use water quality to track flow paths. So for me, the critical zone processes of interest are really the ones that relate to the storage and release of water. And when we try to think about these kind of storage processes of water and release process of water, as well as the biogeochemical patterns that we observe at the catchment outlet, we kind of know that these are quite strongly linked to all these catchment attributes that we can measure. So for example, if we've got 5 catchments and they all differ in their characteristics, they will show different biogeochemical patterns at the outlet. So for example, we may have a smaller catchment and a larger catchment, and they will be different in respect to their biogeochemical patterns at the outlet. Same if we have a steep mountain headwater catchment, or something that is forested, or a catchment that has agricultural land use. So all of these things influence the biogeochemistry and the chemistry that we measure. Likewise, the one important thing that brings us all together here, the critical zone. So everything that's under the land surface, the soils, how deep they are, what type they are, how far it is to the bedrock, all of those things influence catchment biogeochemical patterns that we observe. That's probably no news to anyone. But what we do try to do from these landscape characteristics is try to infer the processes that are actually happening, that are related to the storage and release of water. And then we try to also bring in the biogeochemical processes. So often, I think, and this is, well, where I'm really grateful to the previous speaker, we look at these small-scale processes that we observe in the lab, and then we try to scale them up to the catchment scale, to the landscape scale. And well, I'm not sure, maybe I'm the only one, but I always struggle with this scaling up process because I find it really, really difficult. If there's someone in the audience who's cracked that process, please come talk to me and explain to me how it works. But yeah, because I think, I mean, we talked about, there was talk about heterogeneity in the small scale processes, and they are obviously reflected on the landscape scale, so it's hard to do. But the other thing is also that processes depend on the relevance of these processes depends very much on the spatial scale. So something that we can measure in the lab and in situ maybe in a small soil column or in a soil trench may not necessarily reflect what we measure at the catchment outlet. So that's not to say that these small processes in the soil don't matter. They probably matter for atmospheric exchange, they matter for denitrification in that location and the nitrate uptake, but they may not matter for biogeochemical processes, water quality that we measure at the catchment outlet. So what we have been doing quite a lot, not just me, but I think a lot of people in this room, is actually trying to go the other way. We're trying to observe processes or patterns at the catchment scale, and then from those go back and try to infer the processes that are happening that are relevant for the larger scale, for the larger water quality perspective from the catchment outlet. So we use these observations at the catchment outlet and try to infer the biogeochemical processes that are happening and that are relevant for the outflow. So one of the things, I'm not a biogeochemist, but I work with water quality to track these processes. And the reason why I think looking at the catchment outlet in the stream is useful because the catchment outlet integrates all over that catchment area. So it tells us all the different cumulative processes, what their combined effect is. And what we understand as water quality is actually quite simply, typically just concentration measurements in water samples. So I work in an earth science department, and sometimes I work with colleagues from different fields, and they're always amazed at how simple it is to do water quality measurements. We just go to the stream, take a bucket and dip it in the water, and then we measure what's in that. Whereas deep rock geologists, they do far more sophisticated things, and also yesterday we saw all these dendro approaches. But we use a very simple technique, and it gives us still a lot of information. So we can measure the concentrations in those water samples. If we also know that discharge in the river, we can also quantify the solute loads and solute fluxes of a river, of a catchment. And finally, we can actually look at these biogeochemical patterns in terms of relating concentrations to flow. A lot of you will be familiar with the concentration-discharge relationships. So these are looking at how concentrations vary with flow. The blue ones are so-called dilution patterns. So basically, concentrations decrease as discharge increases. And because we know that at low discharges, rivers are fed by groundwater, we can take this pattern and use it as indicative of a groundwater solute. So saying that these solutes that show this pattern indicate when we have contributions from groundwater. Likewise, the red pattern is a flushing behavior. That's assumed to be solutes that are located in the shallow soil layers. So we are using the observations, again, at the catchment outlet to try to infer these hydrological processes of water storage and water release. We can, of course, take those further. We can, concentration-discharge relationships aren't always straight. So they've got bends in them, and people have looked at those and have said, well, actually, these bends are probably related to biogeochemical reactions taking place in the catchment, or they're related to hydrologic connectivity in the catchments. But basically, it's always the same thing, using biogeochemical patterns at the outlet and inferring the processes that are occurring in the landscape scale. And we have done this extensively on long-term low-frequency data time series. And we kind of understand how this kind of water storage release happens on this long-term scale. And then again, thanks to, for example, Niels for talking about extreme events, this is the thing that I talk about as well. We don't really know what happens to water quality during events. We have observations from long-term data that show that these short-term events deliver huge amounts of, or that the largest amount of solutes are delivered to the stream during those short events. So these extremes don't just have influences on water quantity, but also on water quality. And when I talk, and for all of us who don't think that climate change is a Chinese hoax, we also know that extreme events and generally, generally events will become more important in the next few years. That means we really need to understand what happens at these event timescales. And they don't have to be these massive events that happen once every 100 years, but we have a lot more of these events in general. So if the solute mobilization happens differently during events, then we should have an idea of why and be able to predict that. So really the question is, if we think about these, so these are these kind of long-term, low-frequency concentration discharge relationships, what happens at this end? If we look at events, is it just simply an extrapolation of the known relationships, or does something else happen in terms of solute mobilization that affects water quality in a different way? And the answer is quite simple, yes, there is a huge difference between event scale and long-term water quality. So here you can see in dark blue teal the long-term relationship. This is from a Swiss headwater mountain catchment, and we see kind of these relationships here. But then if you look at the individual events, so some of them are colored here in different colors, you can see that for some of the events, some of the solutes, the event scale water quality dynamics differ substantially from those of the long term. And well, now we can ask what that means, and it's simply that the processes that are relevant in the long-term scale are not necessarily the ones that are relevant at the short-term event scale in terms of water quality. So when I talked about the scaling up, I said that the process relevance depends on the spatial scale, but the process relevance also depends on the temporal scale. And this is often illustrated with the Simpson's paradox, so where I think this was originally done by Simpson on looking at penguin populations, that if you look at individual subgroups, so in our case, these are individual events, you get a different relationship than when you aggregate over all of those different events together. That is not to say that any of those processes are wrong, it's just different processes are relevant at different timescales. And you can also see, when you just look at the events, that one event differs quite substantially from the next event. And we try to, in that paper, link that a bit more to things like event characteristics, so how much precipitation fell, how dry was the catchment beforehand, in a more, like, it's just a very simple correlation analysis. And we found that different solutes seem to be sensitive to different types of characteristics. So a PhD student of mine, Shibangi Cogbrigade, she's currently working on delving a bit deeper into that and trying to understand the mechanisms of that. For that, she's using principal component analysis on the solute data that we have from these sub-hourly event scales. And so what you can see over here on the left-hand side is this kind of is not a CQ relationship. These are just the principal components, well, the first and the second principal component for all these events. They're colored by season, but that's not really relevant for this part of the story that I'm trying to tell here. But if you look, well, with a little bit of imagination, you can definitely see a triangle here, I think. And she, so with a missing bit here, we can talk about what that missing bit means, but and so she tries to trace all the different individual events in this. And what she found is that all of the events start and end in this area over here. And then they take traces up to here or here or here. So whereas the base flow composition is quite well defined, which kind of conforms to our general understanding of how groundwater works as a big groundwater body that's relatively well mixed. We can see that the different peak flow signals, so at the highest flow, so flow is not in this principal component analysis. This is just, that drops out that the chemical signal at these peak flows is quite variable depending on the individual event. And she's looking at currently using stable water isotopes, for example, to try to understand how different contributions from different parts of the catchment, younger and older water, shallower and deeper water, whether we can explain these various signals by looking at these different sources. Additionally, she is also looking at what the influence is of hydrological metrics of these events. So she's again she's done a principal component analysis, this time not on the solute data, but on all the event characteristics that we quantified, things like precipitation intensity, lag times, the runoff volumes, runoff coefficients, and so on. Oh, sorry. Well, I skipped ahead a little bit because we tried to see, winding back, so basically we tried to see that whether this was only for this one catchment in Switzerland or that actually happens elsewhere too. And these are just similar analyses for different catchments in the UK, and we find a range of different patterns. Some of those show the same triangle with base flow concentrations down here, and then again, diverging for peak flow conditions, but others show this kind of very squiggly lines that have no common base flow composition, base flow signal. So it seems to be, again, that's this work in progress, trying to figure out what's happening and why these are so different. But now coming to the event characteristics. So this is a PCA on the event characteristics, like the precipitation intensity and so on, and trying to link those to the solute solute delivery that we are seeing. And what she found is that one of the event dimensions on intensity, for example, so that's precipitation intensity, but it's also the magnitude, the size of the event, so discharge, the runoff coefficient. That is very strongly related, for example, to the peak flow concentrations of all solutes. She also found that flashiness, so how quickly the stream flow increases, how high the peak of the stream flow is compared to the total runoff volume, those things seem to influence the change in concentration for the base flow cations, so sodium and calcium. Antecedent wetness, so how dry was the catchment beforehand, has the strongest influence on sulfate, which is probably due to the geology at that catchment. But basically, what this shows is that there is quite a strong influence on these catchment conditions, on the solutes that are being delivered, and also so how this interaction works between the hydrological conditions, the hydrological mechanisms, and the biogeochemical water quality patterns that we are observing. And we know these sort of things from various different sites. So there are a few quite well-studied catchments all over the world. We saw, those of you who went on the field trip on Monday, saw one of those sites. But we don't really have this understanding across sites. But it would be, well, if we want to be able to predict what's going to happen with climate change, how these events are going to impact our water quality in the rivers for ecological and drinking water purposes, we have to have a better understanding of the mechanisms. And so basically what we've been doing over the last few months, and started a few months ago, is trying to compile data from the UK, from across the UK or across England. So this is the UK with Scotland and Wales cut off here. That's the way it's presented, if you're not used to that shape. And the Environment Agency in England has gotten quite good at deploying high-frequency sensors and collecting high-frequency sub-hourly water quality. The problem there is that there are two different departments in the government that are responsible for water quality and for hydrometrics, so discharge gauging. So it turns out that out of the 200-something sites with high-frequency water quality, only 50 are located in proximity to a gauging station. And we can talk about what that means and why that's the case, but so what we had to do is we had to look at, for sites that were within about 10 kilometers upstream or downstream of a gauging station. It's not perfect, but that's the best we could do. So certainly there are limitations based on that. So that gave us about 50 sites across the UK. There's some tidal sites in this one that were also excluded in the end because of tidal influences. But yeah, so there's a range of sites. We looked at, we characterized the runoff events from these sites. So basically dissected the time series and also looked at things like runoff coefficient, precipitation intensity, and so on for these events and sites. And then we calculated concentration discharge relationships for those events using linear least squares approaches. So one CQ slope for every event and every solute. Now you're going to ask what solutes, and that's a very valid question. Again, this is most likely probably not the solutes that many of us are interested in. So looking at the abstracts, there is a lot of nitrate in the abstract because we care about agricultural processes, right? But that's not one of the key determinants that the Environment Agency in the UK is interested in. So the key parameters they measure is dissolved oxygen, ammonium, chlorophyll, pH, and conductivity. I'm not sure how many of you work with wastewater, but these are the parameters that are of relevance for wastewater. So the reason for the monitoring that happens by the Environment Agency in the UK is mostly for compliance purposes for wastewater treatment plants. So hence we have these parameters. That means we get a lot of input from we can measure the influence of human activities a lot more than we can concentrated human activities like sewage treatment works, but not so much the agricultural inputs. So that's just it's a different direction than other people are doing. But this is, I guess, what is relevant to the UK at the moment. So the time series look kind of like this. So we can see the high variability in stream flow that is reflected quite well in chlorophyll and also in ammonium, but in a different way. So we have these, obviously, data gaps, and dealing with those is actually quite a bit of a challenge, but it is, well, I'll show you some work in progress. It's not finalized yet, but we're getting somewhere. The first thing we did was calculate the Gini coefficient. So the Gini coefficient tells us a bit about how relevant events are overall. So this actually comes from income and looking at how wealth is distributed across populations, but we can also use it for the importance of events. So if our line, it looks at the national income in its original purpose against the total population, and if our line falls on this line, you have perfect equality, and our Gini coefficient is 0. On the other hand, if we have perfect inequality, so one person earns or has nearly all of the wealth, then we'd have this outer line where our Gini coefficient would be 1. And so it's looking at the combined area of AB against the area of A. And we can do that for solutes as well, looking at fluxes and loads when are most of the loads being delivered. And I'm showing you, rather than showing you all the 50 sites, I'll show you 7 sites. These are the ones that we have processed the most so far. And you can see that these Gini coefficients range anywhere from 0.3 to nearly 0.9, are slightly different for each determinant and differ between sites. But in general, we can see that events do play a main role. So in some ways, well, it makes sense to do this kind of research. And then this is an example of concentration-discharge relationships for ammonium and chlorophyll from one of the sites. And you can see that is, so the light gray points in the background are the non-event points. And then we've got all the different events colored here, or at least, every second event colored for illustration purposes only. So yeah, you can see that these are quite diverse and variable. This is also reflected in when we just look at the concentration discharge slopes. So this B value that's plotted here represents the steepness of this slope. A CQ slope value B around 0, like we see for dissolved oxygen, means that concentrations vary independently of discharge. That's not surprising for dissolved oxygen, because dissolved oxygen is known to be more determined by light availability than by flow. For pH, we see mostly negative concentration discharge slopes, so a relationship like that, which is indicative of base cations. And that's kind of what we've been finding at many, many sites, that conductivity is a good proxy for base cations for groundwater contributions. pH is a good proxy for groundwater contributions. The highest variability we see with chlorophyll and ammonium. And so we can see that different sites behave differently. Also, there's a very large variation in these concentration discharge slopes. And the question is, what drives these variations? And for this, I'm going to give you, so this is ongoing analysis, but I think it'd be nice to I just want to show you the first results from those 7 sites. So what we find from an ANOVA is that there's a strong determinant-site interaction. What that means is that every solute at every site behaves differently. And that's basically the ANOVA quantifying what we've been seeing, this complexity that we've been seeing, that has made it very hard to find overarching patterns for when we compare between 2 different sites. But there is hope, and there are possibilities to bring some order into this structure, into this chaos. So we have been finding some general effects. And so these are general effects that are valid for every site, every one of these 7 sites so far, and every one of the 5 determinants we've looked at. And what we've, for example, seen that if the runoff coefficient increases is higher for an event, we likely have a lower CQ slope. So just to remind you, a higher CQ slope can mean less dilution or a stronger enrichment during an event, and a lower CQ slope, like we're seeing for the runoff coefficient, means a stronger dilution during an event or a lower enrichment during the event. Then we also see some determinant-specific effects. So these are valid for all sites, but differ between determinants. And we can see that, for example, if precipitation intensity increases, we see a steeper or a more negative slope for conductivity, which is we see down here. And if precipitation increases, the chlorophyll is more likely to be enriched and flushed into the system. So these kind of conform quite well with other studies we've been doing, where we've been looking at groundwater flows and subsurface flows that are kind of linking precipitation intensity to these flow processes. So this is kind of a different angle, going from stable water isotopes and combining water quality to find similar mechanisms. On the other hand, precipitation intensity seems to have very limited effect on all the other determinants. And then we have some effects that seem to be site and determinant specific. So for example, antecedent wetness or antecedent precipitation results in a decrease in the concentration discharge slope for ammonium and chlorophyll at 2 sites, and at one other site it results in an increase in these concentration discharge slopes. So I think I'm just about in time. So to summarize, we can say that some systems are complex. The water quality dynamics during events are very, very different from the long-term patterns that we can see. And of course, the 7 sites I showed you is only a limited number of sites, but we can bring structure into this complexity if we try. And I think it's worth trying that and worth doing that, trying to link these biogeochemical patterns to the event characteristics and the processes that are occurring. And we have more and more methods and technologies available to measure high-frequency data in remoter catchments that are not too easily accessible with these automated sensors. So I think we should make use of that and try to understand more of what is going on during these events. And with that, I'd like to thank you, and I'm